Hello, I'm Dr. DeBellis. Today I'd like to talk to you about the foundations of psychopharmacology. We're going to discuss the chemical basis of behavior as they relate to prescription drugs. I'd like to also remind you that this lecture is not intended to be a substitute for medical advice. So if you have specific questions about medications that you or a loved one are being prescribed, please direct those toward the prescribing physician. We're going to go over the basics, though, of how psychopharmacology works at the molecular level in the synapse. Neurochemistry focuses on the basic chemical composition and processes of the nervous system, whereas neuropharmacology is the study of compounds that selectively affect the nervous system. Whereas neurochemistry is more focused on endogenous ligands. Neuropharmacology is focused more on exogenous ligands. The last lecture we focused on neurochemistry, but in this lecture we're going to focus on neuropharmacology. We're going to focus primarily on how three different classes of exogenous ligands exert their effect to reduce anxiety, psychotic symptoms, and depressive symptoms. Hopefully you recall from our previous lectures that a ligand is a substance that binds to a receptor molecule where it exerts an effect on that receptor. We have endogenous ligands. Endogenous ligands are produced inside the body, whereas exogenous ligands are produced outside the body. So dopamine, norepinephrine, serotonin, those are going to be endogenous ligands, whereas drugs, whether prescription or recreational drugs, those are probably going to be exogenous ligands. And we're going to focus on exogenous ligands, primarily prescription drugs, in this lecture. So as we previously discussed, receptor subtypes may trigger very different responses to target cells. And drugs can be designed that only affect one specific subtype. We're going to talk about one of these drugs, antipsychotic medications. Believe it or not, they have a particularly strong affinity toward dopamine subtype 2 receptors, which is one of the things that gives them their beneficial effect. So neurotransmitters affect targets by acting on receptors, and receptors are protein molecules in the postsynaptic membrane primarily. We can also have them in the presynaptic, but classically we think of them as being in the postsynaptic. Now, ionotropic receptors quickly open an ion channel when the transmitter molecule binds, whereas metabotropic receptors alter chemical reactions in the cell. They'll send a G protein into the cytoplasm, and it will diffuse throughout the cytoplasm, and it will actually travel across the intercellular space until it hits an ion channel. And once it exerts its effect on the ion channel, then that will actually cause an ion, a cation or an anion, to enter the intracellular space. So we have different subtypes for neurotransmitter receptors. For example, we have 14 different receptor subtypes for serotonin, 5 for norepinephrine, 5 for dopamine. But different subtypes are constantly changing their concentrations in response to all different behaviors. This is one of the things that allows for neuroplasticity. But pharmaceutical drugs exploit the fact that certain drugs target certain subtypes, leading to specific effects associated with that particular subtype. A little review here. Agonists, antagonists, and inverse agonists. As a metaphor, let's assume that a key is intended to lock a door. So we can say that that key that's being inserted into that lock is an agonist. Now, an antagonist, it blocks a receptor. It would be akin to a key that's placed into a door, but the one who puts the key in does not actually lock the door. As a result, it's occupying the space, kind of like a an antagonist that's occupying a receptor, but it doesn't actually exert the effect that one would typically affect that defines an agonist. Now, an inverse agonist is an agonist that actually initiates an effect opposite the normal function. So we can say if you were to put that key into the door and unlock the door, that would be akin to an inverse agonist if an agonist was locking it. It's important that we clarify what binding affinity and efficacy are. Binding affinity is the degree of chemical attraction between a ligand and a receptor. So here we have a ligand, the red ligands that you can see at the bottom of this screen. If they're low affinity, they slowly travel across the synapse. They're not that attracted to the receptors. As a result, they're less likely to activate it. Now, if these were high affinity ligands, we can expect that they would be highly attracted to the receptors. They would go straight to the receptors really quickly. Now, efficacy is the ability of a bound ligand to activate the receptor. If these are low efficacy, as you can see, these ligands are bound to the receptors, but they're not causing graded potentials. If they were high efficacy, we would expect to see graded potentials or localized potentials all over the place whenever they bind to the receptors. So agonists have high efficacy, but antagonists have low efficacy. Partial agonists and partial antagonists are ligands that produce a medium response regardless of dose. So as you recall, an agonist, and here the red circle represents an agonist, it binds to a binding site on the ligand-gated channel. Here's the ligand-gated channel. It's a receptor. And once bound, if it has efficacy, we can expect that there's going to be an opening of the channel, which is going to let an ion in. Now, non-competitive ligands bind instead to modulatory sites on the receptor. So you see how you have these two sites on this receptor, and you have two different types of non-competitive ligands. This is something we see actually when we look at the GABA receptors that benzodiazepines bind to, that we have all different binding sites. So ligands can actually bind to different parts of the same receptor, and they can actually increase the effects of other ligands. Now, a competitive antagonist competes for the same binding site as an agonist. So here the antagonist gets in the way of the agonist. The agonist is not able to bind to the receptor. Now, if these were non-competitive antagonists, they prevent a ligand from binding by activating a different site. So as you can see, we have two different binding sites on this receptor. And whenever the non-competitive antagonist on the left binds to a site, it closes the binding site for the agonist. So the agonist cannot bind, can't exert its effect upon the receptor. A dose-response curve is a graph of the relationship between drug doses and their effects. So here we have an x-axis representing the dose of the drug, whereas the y-axis represents the response, the effect that the drug has on an individual. And you have these two lines here. The green line represents the effect, how effective the dose is. So the effective dose 50 or ED50 helps half the subjects. So this would be the ED50. The LD50 or lethal dose 50 kills half the subjects. Now if we were to place a horizontal line between the ED50, which would be about here, and the LD50, which would be here, the distance between the two would be referred to as a therapeutic index. So it's the distance between the ED50 and the LD50. And safer drugs would have a higher therapeutic index. If, for instance, we were given the example of if an M&M, if one M&M is all it took to make one smile, but 50 of them would cause an overdose, that would be a lot safer than if you needed 20 M&Ms in order to smile, but 21 M&Ms would actually cause an overdose. So we would say in the latter case, it would be a lower therapeutic index than in the former case. Now, drug tolerance can develop. Successive treatments can end up having decreasing effects. We have metabolic tolerance when organ systems become more effective at eliminating the drug. We see this, for instance, with alcohol. Individuals who drink alcohol for the first time will have a stronger response than individuals who are regular drinkers, because individuals who are regular drinkers have a certain level of circulating enzymes that are anticipating that the alcohol will need to be metabolized. Now, functional tolerance is when a target tissue may show altered sensitivity to the drug. We see this, for instance, with MDMA, where we have an increase, or we have a decrease in the number of serotonin and dopamine receptors, or neurons, as a result of subsequent administrations of the drug. Now, changes in the number of receptors can alter sensitivity in the direction opposite to the drug's effects. So neurons downregulate, they decrease the number of receptors in response to agonists, whereas they will upregulate in response to an antagonist. For instance, we're going to learn that antipsychotic medication will antagonize dopamine 2 receptors. As a result, postsynaptic neurons will actually upregulate the number of dopamine 2 receptors that are available for binding. As a result, an individual might need to take a larger dose of antipsychotic medication as a result of the upregulation. And upregulation, again, is in response to an antagonist, whereas downregulation is more likely to be caused by agonist drugs. Now, cross-tolerance is tolerance to a whole class of chemically similar drugs. For instance, an individual who's been taking a barbiturate or a benzodiazepine for an extended period of time will have a higher tolerance to that particular benzodiazepine. In addition, if they take another benzodiazepine instead, they'll also have a higher tolerance to that as well, because it's a class of chemically similar drugs for the two of them. Now, withdrawal symptoms may be caused by drug tolerance, and sensitization occurs when drug effects become stronger with repeated uses or treatments. So the amount of drug that's bioavailable in the body or free to act on the target varies with the route of ingestion. For instance, if somebody takes a drug intravenously or nasally, we'd expect an increased bioavailability than if an individual ingests it orally. And the duration of a drug's effect is determined by how it's metabolized. Biotransformation produces active metabolites that may produce side effects. Some drugs, whenever they're metabolized, they're actually metabolized into active metabolites that have similar effects. This is one of the reasons that many benzodiazepines are longer acting. The metabolite is also capable of having an effect on the body. In some cases, the metabolites can actually be more powerful than the drug itself. We'll find that with Wellbutrin, where the metabolite is actually has more of an effect than the drug itself. Now, postsynaptic receptors can be blocked or activated by drugs, and prolonged transmitter receptor activity can alter behavior. We see this, for instance, when we look at cholinergic drugs like cholinesterase inhibitors. They inhibit the breakdown of acetylcholine at the synapse by reducing the amount of acetylcholinesterase. Acetylcholinesterase, which I abbreviated there ACHE, it's an enzyme that breaks up acetylcholine after it hits a receptor. Probably recall that acetylcholine is a quaternary amine that's involved in cognition, the rest and digest effect, as well as cognition. And whenever we reduce the amount of acetylcholinesterase, we increase the amount of acetylcholine because acetylcholine is no longer being degraded by acetylcholinesterase. So we're going to talk about three classes of drugs, three general classes, the first being antipsychotics. Typical antipsychotics were the first. They're dopamine 2 receptor antagonists. So these are dopamine 2 receptors here, these on the postsynaptic membrane. And the orange circles are dopamine. So whenever the dopamine is released, it should activate dopamine receptors. In this case, these are dopamine 2 receptors. But as you can see, they're not able to because typical antipsychotics antagonize dopamine 2 receptors. So as a result, there's not any dopaminergic activity at these dopamine 2 receptors. Now, atypical antipsychotics came along after typical antipsychotics. They were allegedly better for treating the negative symptoms of schizophrenia, for instance. And here the green circles represent serotonin, whereas the orange circles represent dopamine. And these are receptors on the right for dopamine 2 and serotonin. And if you take atypical antipsychotic medications, the medication antagonizes not just dopamine 2 receptors, but also serotonin receptors. So as a result, serotonin and dopamine are not having any activation on these receptors, and the net effect is diminished dopamine activity, diminished serotonin activity. Now, both the typical and atypical antipsychotics reduce motor activity, and this seems to help alleviate major agitation in many patients. This is the case with people with schizophrenia. We also find that these medications can be useful for other people with agitation, such as people who maybe have had a head injury or somebody who has dementia. Oftentimes a mild antipsychotic medication can help them to reduce their agitation and increase their quality of life. But antipsychotic medications can also give rise to a syndrome we call tardive dyskinesia. It's a reversible syndrome in which an individual will experience tremors and Parkinsonian extrapyramidal behaviors. Now, too much dopamine is associated with psychosis, and generally speaking, too little is associated with movement disorders, such as Parkinsonian symptoms. Now, antidepressants are our second major class of medication that we're going to talk about. We have MAOIs, TCMs, SSRIs, AMs, and SARIs. MAOIs are monoamine oxidase inhibitors. What they do is they inhibit the effect of this enzyme called monoamine oxidase that degrades, that metabolizes monoamines, monoamines being serotonin, dopamine, norepinephrine. Tricyclics are abbreviated TCMs. They were the second major class. And then SSRIs, selective serotonin reuptake inhibitors. We have two newer classes, the adrenergic modulators and the selective androgen reuptake inhibitors. First, we're going to talk about the earliest of the antidepressants, the MAOIs, the monoamine oxidase inhibitors. They prevent the breakdown of monoamines at the synapse. So we have a list of some different ones, Nardil, Parnate, Marplan. But basically, they have the same general effect. What they do is they prevent the breakdown of these monoamines. So the orange circles, again, are going to be dopamine. The purple ones are going to be norepinephrine, and the green ones will be serotonin. And monoamine oxidase, which we're going to find in the postsynaptic neuron, what it does is it breaks down the dopamine, norepinephrine, and serotonin, because these are monoamines. So here we find activation of the receptors, these local graded potentials, and then the monoamine oxidase rushes out to the receptors where it will actually degrade the norepinephrine, dopamine, and serotonin. As a result, there's less of it available in the synaptic space. Now, if this patient was on an monoamine oxidase inhibitor, we would expect that, and here the monoamine oxidase is represented with these puvules, these white puvules here. And what they do is they block the destruction of the monoamines by monoamine oxidase. And as a result, we find that there's renewed availability, bioavailability of the dopamine, norepinephrine, and serotonin. So the long-term effect of the MAOI is an increased availability of unoxidized monoamines in the synaptic cleft. So by preventing the breakdown of the monoamines, this is what we have, an accumulation of monoamines. Prolonging their activity is a major feature of antidepressant medication. The second class that came along was the tricyclics, and below you have a list of different tricyclic medications. But tricyclics prevent the reuptake of amines into presynaptic terminals. So the next class is going to be selective serotonin reuptake inhibitors. Tricyclics are actually non-specific. They actually work on different neurotransmitter receptors. So they prevent the reuptake of amines into the presynaptic terminal. They prevent specifically the reuptake of serotonin, norepinephrine, and acetylcholine. They also antagonize certain androgens and histamine, but we're going to focus on the serotonin and the norepinephrine pathways for the purpose of this lecture. So the tricyclic antidepressant will increase the norepinephrine and serotonin by blocking their reuptake. So we're adding a new component to this particular diagram. Here we have this black rectangle. And what this is going to be is it's going to be a transporter, a membrane transporter that actually transports neurotransmitters back into the presynaptic terminal. So again, the orange circles are dopamine, the purple are norepinephrine, green serotonin, and we'll also see monoamine oxidase, which is red. So as you can see, these monoamines are released into the synapse where they diffuse across and they hit receptors. And then when they hit the receptors, some of these monoamines are actually going to be transported back into the presynaptic terminal, where they're then inserted into these vesicles for their own protection. Because remember, if you're an amine, you want to be in a vesicle because that's like a lifeboat that protects you from these free-floating enzymes in the cytoplasm that will actually destroy them. But the different reuptake transporters that are affected by the tricyclic medication are SERT, DAT, NET. Those are serotonin reuptake transporter, dopamine transporter, and norepinephrine reuptake transporter. But they all pump these monoamines into the presynaptic neuron, and they reduce their bioavailability in the synapse. Now, if the patient were taking tricyclic medication, we would expect to see an increase in the monoamine bioavailability. And here we see the inactivation of the transporter. And as a result, we end up with an abundance of these neurotransmitters. So we're going to have heightened activity due to the bioavailability. We're going to have heightened activity for serotonin, norepinephrine, and dopamine. Now, contrary to initial conceptions of depression, we've learned that depression's not just about serotonin levels. It's also about dopamine levels. It's also about norepinephrine levels. Dopamine's really important, for instance, for motor functioning. And as you're probably aware, one of the symptoms of depression is psychomotor retardation. And that's probably related to dopamine levels. Norepinephrine is another neurotransmitter that is involved in depression whenever it comes to the fight or flight response, whenever it comes to energy levels. Clearly, these are things that are going to be involved in depression. So here we can see there's an increased bioavailability of the dopamine, norepinephrine, and serotonin. Now, SSRIs, or selective serotonin reuptake inhibitors, are a third class. These are very commonly prescribed. They're safer to prescribe than tricyclics. And like Prozac or Zoloft, they allow serotonin to accumulate in the synapse with fewer side effects than tricyclics. So these allowed us to isolate serotonin and increase the level of serotonin, to selectively increase serotonin level by means of inhibiting reuptake by this transporter in the membrane that we're going to call SERT, serotonin reuptake transporter. Here are some SSRIs that are in use. And many of these SSRIs actually also help with things like anxiety, and each one has sort of a specific condition that maybe it's most useful in helping with patients. But when serotonin's released in the synapse, if somebody is not taking an SSRI, we would expect that the serotonin there, the green circles, are going to diffuse across the synapse, and then they're going to be pumped back into the presynaptic terminal. Some of them are actually going to be destroyed by monoamine oxidase, which we talked about earlier. But they're pumped into that vesicle where they're safe from those enzymes that like to eat up serotonin. Now, if the patient were taking an SSRI, the inactivated SERT, or serotonin reuptake transporter, would create an increase in the bioavailability of serotonin in the synapse. So here we see, let's get rid of that reuptake transporter. There we go. It's gone. And as you can see, after the serotonin hits the receptor, it actually is not reuptaken. So we end up with this congested, highly bioavailable serotonin level. So monoamine oxidase is still going to destroy some of it, but you have a lot more serotonin in the synapse because the serotonin is not being reuptaken into the presynaptic terminal. This would be akin to if you had a basketball team and you were supposed to take a player out of the game before you substitute another in, if you were allowed to just leave 6 players in the game, maybe 7, 8. Over time, we're going to see there's going to be much more activation in the serotonin pathways as a result of this increase in serotonin levels in the synapse. Now, selective serotonin reuptake inhibitors can also cause certain unwanted side effects. Serotonin syndrome would be an example when somebody has too much serotonin. They can also stimulate the central nervous system, affect reproductive functioning, and they can be associated with insomnia. So these are some of the side effects. But generally speaking, they boast less side effects than many MAOIs and tricyclic medications, which makes them in some ways easier to prescribe. Now, AMs, or adrenergic modulators, they exert their effects on norepinephrine and dopamine pathways by blocking dopamine transporter and norepinephrine transporters, which are both in the presynaptic membrane, thereby preventing the reuptake. The classic example of this is going to be Wellbutrin. And the way that Wellbutrin is going to work here, the purple circles are going to be norepinephrine, the orange ones are going to be dopamine. And we find that the dopamine transporter in orange is going to be on this presynaptic terminal down below, and up top the norepinephrine transporter in purple. But you can see these vesicles are releasing dopamine and norepinephrine into the synapse. And normally we would find that those neurotransmitters would activate the receptors, causing these graded potentials. And afterwards, reuptake by the transporters would take these neurotransmitters and then they would repackage them safely into these vesicles for further use. Now, if the individual were taking Wellbutrin, we would expect norepinephrine and dopamine reuptake transporters to deactivate and create an abundance of both dopamine and norepinephrine in the synapse. So here we see the removal of both of our membrane transporters, and whenever the dopamine and norepinephrine is released, it causes these localized graded potentials, but in addition, it's not reuptaken. So we ended up with this abundance of norepinephrine and dopamine in the synapse. So the bioavailability increases, and many people report that they have less depressive symptoms whenever they take a medication such as this. So you're going to have more localized potentials. Now, researchers discovered the drug bupropion or Wellbutrin actually gets metabolized into an active metabolite that's more powerful than the drug itself, suggesting it's actually a form of precursor therapy. In our final class, the serotonin reuptake inhibitors, such as the serotonin receptors, while also preventing serotonin reuptake. And our final class is going to be serotonin 2 antagonist and reuptake inhibitors. These are going to be drugs such as trazodone and surzone. You're probably familiar with trazodone. Trazodone seems to have a lot of benefits for promoting sleep in those who are depressed with insomnia. But these work as antagonists of serotonin 2 receptors while also preventing serotonin reuptake. And that's our fifth class of antidepressants. Now we're going to move to our final topic, which is sedative hypnotics and anti-anxiety agents. Anxiolytics or tranquilizers or depressants. They're drugs that reduce nervous system activity. So these are going to be used for people suffering from conditions like anxiety. Now, benzodiazepine agonists are also known as minor tranquilizers, and they act on GABA A receptors. GABA stands for gamma aminobutyric acid. We're going to call it GABA. But GABA receptors have several binding sites that enhance or inhibit GABA's effects. And benzodiazepines, they bind at an orphan receptor. We're not sure of the endogenous ligand. A possibility is allopregnanolone, which is a steroid elevated during stress. It's calming. But other neurosteroids may also act on these GABA A sites. But the benzodiazepines exert their effect on GABA pathways, thereby polarizing postsynaptic neurons. So here we see the green circles are going to be this exogenous ligand, and they are going to affect these GABA sites, thereby actually causing these local potentials. They're going to cause a massive influx of chloride. Anytime these receptors are activated, chloride is going to hyperpolarize the postsynaptic neuron. And they work as non-competitive ligands that bind to a modulatory site. So red's going to be GABA, and the green's actually going to be the benzodiazepine. And as you can see, whenever it activates the binding site in conjunction, the GABA is actually going to open the ionotropic receptor. And what we're going to actually find is an influx of the chloride, which results in a hyperpolarization of the intracellular space, which makes the neuron less likely to fire, which is not surprising when you consider that most drugs that have GABAergic effects are going to decrease activity in the central nervous system. So, thank you very much. Now we've gone over the fundamentals of what happens in the central nervous system when somebody takes psychotropic medication. Please tune in, and next we're going to go over drugs of abuse and how they affect the nervous system and exert their effects. So thank you very much.